everyone. My name is Kiera Davis, and my presentation is Go with the Glow, also known as Plant Glowing. So, just get y'all minds thinking a little bit. As this picture shows, we live in an unsustainable world using resources faster than we can replenish them and pushing the earth beyond its fragile limits. Nature shows us a better way as biology is sustainable so we can harness genetic engineering to create better solutions. So the history of glowing plants started with bioluminescence. The importance of luminosity has resulted in lots of research into an improvement of bioluminescent systems. So a brief history of glowing plants. The first glowing plant was created in 1986 with the help of luciferase luciferin gene. Then a gene was added to make glow by luciferin. In 2010, genes were added to make luciferin that result in plants which glows dimly. The University of Cambridge created a glowing bacteria by recycling luciferase, which is an enzyme. So, as I mentioned before, the first bioluminescent plant was made in 1986. It requires 8 hours of exposure, and the color of the glow in plants is determined by the luciferase. So here's a video made by the biotech company which creates autoluminescent plants.
Glowing animals and plants seem to be all the rage these days. A biotechnology company based out of St. Louis recently unveiled a commercially available plant that is entirely autoluminescent. For years, Dr. Alexander Kruszewski, BioGlow's founder and chief scientist, has been studying the molecular biology of plants in addition to the bioluminescence of aquatic bacteria. All the hard work paid off. His newest creation is being referred to as the world's first autoluminescent plants. While glowing plants have been introduced in the past, the way in which they derived that lighting effect was due to chemicals and UV light. BioGlow's offering gets the glow from the undergrowth of the plant. DNA forms found in nature are utilized, similar to the type of light fireflies emit. Said a spokesperson at BioGlow, the autoluminescent plant's light emission machinery is encoded on a cellular level, allowing the plant to constantly emit visible light during its life cycle. Kruszewski's goal in developing the plants is to develop more sustainable, cleaner, and affordable light sources.
So I know I have used a bunch of terms that you may know the definitions to. Luciferase, which scientists insert the gene for luciferase along with a promoter, and then the luciferin is then manually added. So luciferase is an enzyme which is formed from the luminescent organism, and we all know what a promoter is, which is the beginning of a gene that tells the cell to start the transcription to produce a protein. And luciferin is a chemical that produces light when oxidized. So, and also, these glowing plants have also been seen in the International Space Station as well. So the plant that is used, or the plants that they sometimes are used, is the Rabidosus, which it is a small flowering plant and is the member of the mustard family. It is like the fruit fly of plant biology, and it's also used because it has a short genome, so which it grows at a much faster rate than other flowering plant does. So genes used to create the glow. In the picture on the left hand side shows fluorescently stained Vibrio fishery cells, which is a marine bacteria. It has natural inhabitants of seawater which are found in freshwater, and it has characteristics of bioluminescence which are controlled by a small set of genes known as the Lux operon. So here are the process of creating a glowing plant. First, you transform the plant, and then it's iterated and designed, and then it's sent off for printing. So the software that was shown in the picture before is called the Genome Compiler. So it allows scientists to search for gene sequences and modify those genes using a graphical interface user. So the software is then used to look up Vibrio fischeri genes that code and optimize, that basically adjust the sequences, and then use plants instead of bacteria. So the DNA is then synthesized and ready for print. Then it's then shipped off to a company who makes the DNA. So when the DNA comes back, it goes through two steps. First is inserting into some bacteria, also known as agrobacterium. So the bacteria is then inserted into the DNA of female gametes of the plant. And then second, a gene gun is used, which then fires out the DNA at a high velocity into cells of the plant. So some of those cells will absorb the DNA, and then eventually it will start to express it. So for the recent advances, for the field of synthetic biology, as I talked about the Genome Compiler software, and they also have the Cambrian Genomics. As the chart shows to the right, the cost will continue to fall, but within a few years, thousands of DIY bioenthusiasts will be using citizen science to inspire their own creations. So near on in the future, you will be able to make your own plant. It's not going to glow a lot, but it will show glowing very dimly. And Cambridge Genetics is their first commercial hardware and system for laser printing for DNA. So here are some advantages. Lights, of course. In the near future, it then may replaces the lamps, which also can save money, which we all need. So it saves electricity, increases oxygen levels. But some disadvantages it can have is very expensive. It's $100 a plant. It may also cause problems to the organisms that are around in our universe, and it also may be harmful to the plant. So is this legal? So there are 3 federal agencies which regulate genetically modified organisms in the United States, and these include the USDA, which regulate plant and agriculture. Then second, you have the EPA, which regulate new uses of pesticides such as GMOs or other pesticides that we use in the world today for the resistance of plants. And then you have FDA, which regulate food and feedstock. So here are my references. I hope that you all learned a little bit more about plant glowing. I never know this existed, but I have learned a lot. And thank you.